Today I'm going to talk about our recent discovery of a nucleic acid-based adaptive immunity in the insect Drosophila melanogaster. So as you may know, to combat viral infection, higher vertebrates have evolved, in addition to their innate immune response, a more complex adaptive immune response that is a protein-based system characterized by an acquired specificity through protein-protein interaction between antibodies and antigen. This immune response and this immune signal can be spread in different tissues through the B and T cells, and most importantly also the formation of a genetic memory of this encounter between the host and the pathogen. But that's just for the higher vertebrates. For the rest of the living organisms in the world, actually they utilize a different specific antiviral response that is mediated through RNA interference, or RNAi. And in this system, the specificity is acquired through RNA-RNA interaction, based complementarity between the sRNAs loaded onto Argonaute proteins that can then recognize the messenger of the RNA. At the molecular level, upon infection, here RNA virus, the replication intermediate is recognized by a dicer enzyme that can cleave the double-strand RNA molecule into small sRNA duplexes that are then loaded onto Argonaute complex and can specifically target the messenger of the RNA. But this is essentially a intercellular mechanism. So how do you get spread of this antiviral response? Well, in plants and C. elegans, RNA signals can spread between cells through specific transporters and channels. And then at the local level, each cell is going to be able to amplify that response. And to do that, what happens is they receive these new sRNAs, and when the Argonaute protein recognizes its target, it recruits RDRP, so RNA-dependent RNA polymerase, and then load onto the targeted RNA and generate new sRNAs, or secondary sRNAs. In flies, we showed a few years ago that there is also spread of an efficient antiviral RNA signal through the endocytic pathway. But it's been known that there is no RDRP polymerase in insects, which raised the question of like, how do flies maintain these systemic antiviral RNA signals, right? Because if you can't make more, how do you maintain that signal? So in the absence of genetic evidence for amplification, I decided to look for the signal itself. Here, the secondary sRNAs. So how to distinguish these two? Well, primary sRNAs, as I said earlier, are produced by enzymatic cleavage, by a dicer enzyme, generating new small sRNAs that are characterized by the monophosphate group at their 5' end. But if you make new sRNAs, then these new sRNAs are actually produced by polymerase and therefore have triphosphate group at their 5' end. And actually that precludes their cloning by normal approach of small RNA sequencing. So I developed a new technique that I call tripRNA-seq that can specifically capture these small RNAs with triphosphate group at their 5' end. And here is what you get when you look at the small RNA population using these two techniques. So you get your flies, you isolate a small RNA, and you do in parallel either normal small RNA-seq or tripRNA-seq. And as you can see here in the x-axis, using the normal small RNA-seq, you mostly clone the microRNA. We have a 5' monophosphate group. And when you use tripRNA-seq, you mostly capture a small RNA that have triphosphate group at the 5' end. Here, the endogenous one coming from the transcriptional start site. So they are your naturally occurring triphosphate sRNAs. So I went back to my flies, injected them with my favorite arbovirus, Synbis, waited a few days, extracted the small RNAs, proceeded with these two techniques, and then look at the sRNAs mapping to the viral genome, right, to see which one will target the virus. And as you can see here, when you look at the enrichment of tripRNA-seq versus normal sequencing, here in gray, you can see that there's a specific, significant and consistent enrichment of vsRNA with triphosphates at 4 days post-infection and also 7 days post-infection. And when you look at the size distribution of these sRNAs, you can see that here in white, the triphosphorylated small RNAs have the same size distribution than normal sRNAs, which is, of course, 21 nucleotide. And they also map from the sense and the antisense strand of the virus. So these first data indicated that there is de novo synthesis of virus-derived sRNAs during infection in flies, production of secondary sRNAs. Okay, that's great, but is it the same thing as in plant and nematodes, which is actually this amplification is specifically triggered by the Argonaute protein, so it's really specific to the response. So to test this hypothesis, we repeated the same experiment in Argonaute mutant flies in gray. And what I found was actually when you do that, here in the wild type, you have this enrichment for triphosphorylated vsRNAs, But when you look in Argonaut mutants, you completely abolish the production of secondary VSRNAs. And furthermore, I assessed whether or not this was specific to viral infection. And so I just injected double-stranded RNA against GFP in flies or green flies expressing eGFP everywhere. And when I looked at the small RNAs deriving from GFP, I couldn't detect any secondary siRNAs in the absence of infection. So showing that RNA amplification requires RNAi targeting by Argonaut and viral infection. So in other systems, I say like the amplification happens in all cells. However, in insects, they're like us, kind of. They have circulating immune cells, namely the hemocytes. You can see here hemocytes feasting off some green labeled E. coli. So we wonder maybe these cells would be directly involved in this response. So we use the genetic tools available in flies to specifically kill the hemocytes by expressing of apoptotic factors, or specifically knocking down Argonaut in these hemocytes. And what we found is shown here in the control, when you map all the triphosphate VSRNAs to the viral genome, here the coordinates, you can see that you have like nice coverage in the wild type in black. But if you kill all the hemocytes, or specifically knock down AGO2 in the hemocytes, you completely lose the production at the organismal level of these secondary siRNAs, just by targeting the hemocytes. So these siRNAs are made in the hemocytes. But again, no RDRP in Drosophila, so how could that be possible to make new siRNAs? Well, a few years ago, Carla Saleh showed that actually viral cDNA could be generated by endogenous retrotransposons in insect cells when they were infected. So converting this viral RNA into viral DNA. So I went back to my flies, injected them, and looked for viral DNA coming from the RNA virus SynBis. You can see here, if by PCR you don't detect any SynBis in the carcasses or in the hemocyte fraction. But if it's infected with SynBis here, you can see that only in the hemocyte fraction can you detect viral DNA being produced. And to confirm that it was specific to RT activity, I injecting the flies with AZT. And you can see here that when you treat the flies with AZT, you don't produce viral DNA in the hemocytes. And furthermore, if you repeat the experiment in AGO2 mutant flies, or flies with hemocyte here are knocked down for AGO2, you don't get the viral DNA. And then to confirm that this viral DNA was used to make this new siRNA, so it was a template actually, and not RNA, but DNA, the template for these new siRNAs. When you treat it with the AZT here in red, you can see that by both cloning approach, you completely abolish the production of new siRNAs. So indicating that the production of viral DNA in hemocytes is required for RNA amplification. Okay, so it's great. We have these cells, these hemocytes, these circulating hemocytes. They can make viral DNA. They can make more siRNAs. But how are you going to protect the fly if only one cell type makes them? So you have to spread the signal. So a good candidate were exosome vesicles because they were already known to carry microRNAs. So we looked at, we used the same genetic tools to target two genes known in flies to prevent the production and secretion of exosomes, specifically in the hemocytes. First, so you can see that when you isolate the exosomes from infected flies and do deep sequencing, you can see here in gray, in the control flies, that you get specifically in the circulating exosomes, siRNAs mapping to the virus. So these vesicles, circulating vesicles, contain vSRNAs, siRNAs against the virus. But when you repeat that experiment in flies with specifically hemocytes cannot secrete exosome, then you don't see that, right? And here's like showing that actually in all situations where you suppress exosome secretion, specifically in the hemocytes, or knock down AGO2 or prevent production of viral DNA, you don't get vSRNAs secreted in the exosomes. All right, so we have circulating vSRNAs, but do they do anything against the virus? So we did this experiment where we went back to our flies. These are not infected flies, or flies infected with SynBis, flies without hemocytes or with hemocyte could not secrete exosomes, infected them, isolate the exosomes, and then treated cells that express the SynBis GFP. And as you can see here, only cells culture that were treated with exosomes from infected flies could prevent spread of the virus in the culture. And we did the same thing in flies. So you isolate these exosomes from infected flies. If the flies were not infected prior extraction of exosome, here in white, the control, right, you don't get protection. But if you infect these flies with SynBis, with exosome from infected flies, then you get strong protection. But if the donor flies were not able to produce exosome, then you don't get protection. So showing these circulating exosomes containing vSRNAs can actually be antiviral and confer passive immunity. So in conclusion, I think I showed you today that there is a bona fide nucleic acid-based adaptive immunity in Drosophila that is conferred and mediated by hemocytes. So the hemocytes can get this specificity through the interaction of siRNAs. They can spread by secreting new siRNAs into vesicles and create a genetic memory by making viral DNA specifically against the virus encounter. And with that, I would like to thank a member of the Andino lab, Raul, who's a great PI, and a former member of the lab. I encourage you to go see Mark Kinotomi and Leonid Gitlin, who have posters today, and former member of the Weissman lab who helped me with this cloning, and Julia Pack, whose work was seminal in RNA amplification in C. elegans. Thank you.
It's time for questions. Over there.
Very nice talk, Michel. I have two questions, one technical and one non-technical. The first one, that non-technical, you think the microRNAs depend on another RNA polymerase than the secondary siRNAs?
So
for their transcription from DNA?
Yes, so you make DNA, and then out of that DNA, you make a new strand that's going to be processed into new siRNAs.
Okay, right. And microRNAs need another...
Oh, I don't see an amplification of microRNA.
Okay.
So that's the thing. It's only siRNAs deriving from the virus. So if you look at other small RNAs, like microRNA, they are not triphosphorated. If you look at piRNAs, so small RNAs that target transposons, they are not amplified either. They're not...
Okay.
And you don't see them in the tripRNA-seq data
Which
set.
means they're transcribed by another RNA polymerase.
So the microRNA, you won't see them, right? Because the precursor is like a hair loop, and so after enzymatic cleavage, you only see the bona fide microRNA, not the pre, right? So you only get this monophosphate. Mm-hmm. And for piRNA, it's the same, where it's actually cleaved into several
And do you think if you isolate the nuclear microRNAs that are not processed by DROSHA, you would see the triple phosphate or no?
So first, and actually, back to your question. So I don't know what the polymerase is. So it could be the same. It could be POL2 that makes microRNA, and it will be the same here. We don't know what's actually transcribing. And then I don't think it will be DROSHA, maybe dicer or another enzyme, because for VSRNA, it's usually a long double-stranded template. So it's usually dicer2 in that case, and not DROSHA that makes the microRNA.
My second question, which is technical, how do you isolate EV? And when you isolate them, how do you make sure you don't have virus? And therefore, this is what confers immunity.
So for the EVs, I do kind of the same like I do for hemocytes. So I extract the hemolymph. I do multiple centrifugation. Then I did two techniques. One is the ultracentrifugation approach with a sucrose caution. And the other one where it's like you do multiple centrifugation and then you precipitate. And at the end, when I have that, I've done qPCR and I don't detect the virus in the exome fraction. Okay. And I've done also where I actually isolate the hemocyte by FACS, and I don't detect the virus in the hemocytes. So they do produce, and actually another thing, it's one of my favorite things actually, is if you look at these new siRNAs, they're the first species of small RNAs that you find in the hemocytes. So like the most abundant secondary siRNA that you can find in the above new transcripts.
Thank you.
How do the ego-negative flies deal with the virus? Do they die?
They're hypersensitive to viral infection.
And is there any differences between the exosome negative and the hemocyte negative? So to see, is there any component of this pathway that is more important or less important, or are they all severely affected to the same degree?
So what I can say is actually it's interesting. If you do, let's say if you do AZT treatments to prevent viral DNA production in AGO2, you don't change the phenotype. So basically you're maxed out in terms of viral replication. Mm-hmm. So it's like showing that, suggesting that it's in the same pathway, right? Like if you suppress AGO2 and then you suppress RT activity, actually you don't increase the phenotype. It's you're already the same. And in wild type, if you just treat with AZT, actually you usually get to the level of AGO2 mutants. Yeah.
Okay, good.
After amplification, do you know what signals the VSRNAs for packaging into exosomes?
Excuse me, sorry?
I said after amplification, do you know what signals the VSRNAs for packaging into the exosomes? Like,
no. How they get packaged into the exosomes?
Yeah, like is there a signal sequence that?
No, I don't know. And what's very interesting is I actually think it's very specific in the sense that if you look at the exosome, so if you look at the fly, you actually can see, so you can see the signature of dicing, right? So dicing is you cleave double-stranded RNA and you make this like specific with like the 3' overhang of like. But if you look in the exosomes, although you do see the same ratio of sense and antisense, you don't see any dicing signature. So they must come as like single-stranded already. And actually, if you look in the hemocyte compared to the rest of the fly, same thing. The signature of dicing is very low. So there is something specific, which I think is actually, we can discuss that more, but which is more akin to the production of pRNAs, the phase production of pRNAs against transposon. So we think it's actually operating this in a very similar manner. That's more for RNAi geeks.
Okay. If there's no more questions, please join me to thank Michel and all the other speakers for a great session.